So viruses are comprised of a capsid, a protein coat that surrounds their genetic information, which can be DNA or RNA. They may have an envelope, which is a cell membrane that is derived from their host, or they can be naked and not have a cell membrane surrounding them. Now, many viruses may also have spikes that protrude from the viral capsid, or if present, they can protrude from the viral envelope as well. So these spikes are basically glycoproteins, so a carbohydrate combined with a protein that kind of extend off the surface, and they can help viruses to attach and gain entry to host cells. And that's because these spikes only bind to specific factors on a given host cell. The spikes pretty much play an important role in determining what species and the tissues that a virus can infect because of that recognition between the spike and the receptors within the host. So the host immune system may also recognize spikes, and they can mount an immune response against them. And in many cases, though, the spikes then change. Once the immune system starts to recognize a certain spike, then the spikes then the virus can evolve and the spikes can change. So some viruses do rapidly and routinely in their in their evolution kind of change out their spikes, and therefore then the new variant escapes immune detection. So influenza viruses, let's say for instance, frequently mutate and experience small changes in their spike proteins. And this is such an important issue, especially for influenza A, that its spike proteins are part of the virus subtype naming convention. So because there's so many different types of spikes in influenza A, it's become part of its name. So for example, swine flu is also what we call H1N1. So it's part of its subtype, while bird flu is called H5N1. So the H and the N refer to different types of spikes. So there's the H is the hemagglutinin spikes, and there's about 18 different variations of these H spikes. And then the N glycoproteins are the neuraminidase, and there's about 11 different variations of those. So there can be a wide variety of different types of spikes, and it can be important in the identification of some of these viruses. Now, I also have down there that a virion is a fully formed virus that can infect a host cell. So sometimes you'll hear virus and sometimes you'll hear a virion. So the virion refers to one that has all the parts and is ready to infect another cell. Now, we talked about the genome a little bit and the nucleic acids. So we said that they can be DNA or RNA as part of the nucleic acids. So the viral genome itself is very small. Really, it only needs genes to encode for how to make the capsomere proteins, right? Because that's unique for that virus. It has to be able to replicate its protein, so it carries the information on how to make those proteins. And there could be some other enzymes that are needed for viral replication or for different structural factors. But overall, viruses don't need a lot of genes. So most viruses have fewer than 300 genes, so the very small genome there. Now, cells can have, as we said, DNA or RNA, but not both. So we can have DNA viruses, and these DNA viruses can be single-stranded, which seems kind of unusual for us because we're used to talking about double-stranded DNA, kind of a DNA helix, right? But DNA can also be single-stranded in viruses. They can also be linear, like we see in eukaryotes, or it can be circular, like we see in bacteria. So a lot of variation then in just the DNA structure, and that's why this is used as a means of categorizing the different viruses. Then you can have RNA viruses, and these can be double-stranded, DS for double-stranded, but most RNA viruses are going to be single-stranded. So once again, we normally think of RNAs being single-stranded like messenger RNA or transfer RNA. We don't usually think of it as being double-stranded, but it is possible to have double-stranded DNA. Okay. And then there's also what's called positive sense RNA or negative sense RNA. And I don't want you to spend too much time because you can these can be kind of confusing, but here in this chart you see single-stranded positive sense, single-stranded negative sense, right? So it just has to do with whether it's ready to move forward or whether it needs to be converted into a different form before it can move further. So positive sense, it's ready, as it says, for translation because it is RNA. So you think of it's already been transcribed and now it's ready for translation. If it's negative sense, it has to be converted first into a messenger RNA form that can then be translated. Okay, but once again, just don't worry too much about them. Just recognize that there's a positive sense and a negative sense. More importantly, though, there are what we call retroviruses. And these guys tend to carry their own enzymes to create DNA out of RNA. So it's an RNA virus and it produces DNA from the RNA. So normally we're talking about RNA being made from DNA. So we transcribe a gene from the DNA and produce RNA. In this case, we're going backwards. We're going from the RNA to making a DNA strand, and that's why it's called a retrovirus, because it's kind of going in reverse. And we'll talk about some of these retroviruses later on. All right. But these can be kind of devious because they're making DNA, so therefore they can insert the viral DNA into your genome. Now, viruses can infect every type of life on Earth. However, a given virus, though, will exhibit a specific host range or a specific collection of species that it can infect. So some viruses infect more than one species, while others infect only one species. So, for example, the measles virus is only able to infect humans. And most of the viruses that infect humans typically are able to infect some other animal. And that's because most human viruses evolved in other species, and then through genetic changes, they gained the capacity to infect us. So, for example, the avian influenza H5N1, which primarily infected certain waterfowl species like geese and ducks, it underwent genetic changes that expanded its host range, so now it includes humans. And the same holds true for swine flu that initially infected pigs, and now it can infect humans. So we consider this to be the host range, right? So the range of organisms that a virus can infect. It can be limited or it can be pretty broad, right? So we have the example here also of rabies, which has a host range of only being able to infect all mammals. Now, viruses can also exhibit specificity regarding what tissues or cells they will infect within their host, and this is called tropism. So tropism is due to the virus surface factors and only being able to bind to specific molecules on certain host cells. So tropism is basically the type of cell or tissue that the virus infects or can infect. For some viruses like Ebola, they can infect a wide range of host cells or tissues and are said to have a very broad tropism. Other viruses have a very narrow tropism and may specifically target only one type of cell or tissue in that given host. For example, the hepatitis viruses infect the liver, so typically they're described as hepatotropic. So getting that tropism and referring to the liver, hepatotropic. There's human T lymphocyte viruses that get their name as a result of their for having partiality for infecting human T cells. So tropisms refer to the cell or the tissue that the virus likes to infect.